Here we are, hooray! Here is an example, yay! Example of a density front. From where is it? It's from the Western Mediterranean Sea. All right. So what's shown here are two vertical transects. So what is a vertical transect? For those of you who are still confused about it, you think about you have a cake. Okay, you have a salty cake, different salinities. So salinity is the salt concentration, and so on. You can cut through the cake. So it's your birthday. You're the first one cutting through the cake. You open it, and you can see the structure of the salinity in terms of different colors. So you allocate colors to different salinity. Okay, you can indeed use also contours to show these profiles. And this is the top one. It's a salinity decay. Okay, you cut through it. And I mean, it would be also good to see the temperature structure, but salinity is good enough in this example. And you can see a front, you can see the change in salinity up here. Yeah, I activate the mouse. Let's see here in this zone. You can see on one side it's all blue. Okay? On the other side, you can see it turns into red type of thing. Okay? So this is the density front. All right, it's a horizontal change. Good. So let's have a look at the bottom distribution. That's another cake. So you cooked two birthday cakes, and what you can see here is so-called fluorescence. And fluorescence indicates the chlorophyll A concentrations. It's a proxy of chlorophyll A concentrations. And there what you can actually see is you can see the downwelling. And the downwelling, you can see that because it's higher chlorophyll A concentrations near the surface. Okay. And then you can see the downward movement down here of higher concentrations and then leading to a tongue-like structure. Okay, so you can see this, the export of water coming from, or that is formed along the frontal axis. All right, you can't see it in the salinity field because the movement is actually along the lines of the same colors, of the same density, of the same salinity. All right, this is frontal downwelling, and here you can see a clear example of density. This itself, this mechanism, is not the productive part of it. Okay, it might be beneficial for some zooplankton species that actually vertically migrate. So they actually can go from the surface to a depth of 200 meters and take advantage of this subsurface chlorophyll signature. But okay, let's have another look at it. Let's have a look at your fluorescence cake, and you can actually see that the entire zone of the front of a relatively wide region, and it's half a degree, and half a degree is something like 50 kilometers, is relatively productive, has high levels. So this is all in purple here. And this is actually a signature of the eddies that are formed in this. But what is an eddy? Eddy is a circular feature, and some eddies can produce upwelling. And you can, for example, see, and one upwelling is actually included in this example. Can you see it? Have a look at your salinity cake, and it's a little bit salt anomaly here up in this at the top in this region. Okay, it's a little bit an anomaly here, and you can also see it in the fluorescence. And this is the signature of the eddy, and the eddy mix horizontally. So you get actually a broad zone of where you have elevated chlorophyll A concentrations, which is a signature of a plankton bloom. All right, so this was one example of a density front.